Hey guys, welcome to Proactive Prudence. In this video, we'll be studying about physiology, and the chapter we'll be focusing on is Introduction to Cell Physiology. So, without wasting time, let's move on to the contents that will be dealt in this video. So, we'll be studying about different cell organelles like cell membrane, mitochondria, lysosomes, peroxisomes, cytoskeleton, centrosomes, cilia, cell adhesion, intercellular connections, gap junctions, nucleus and related structures, endoplasmic reticulum, ribosomes, and Golgi apparatus. Firstly, we'll see what is cell membrane. So, cell membrane is the layer which surrounds the cell. It is made up of lipids and proteins, and it is semi-permeable. It allows certain substances to pass through it. So, it is usually referred to as plasma membrane also. And the thickness of cell membrane is 7.5 nanometers. So the lipid bilayer, that phospholipid bilayer that we see, is made up of phospholipid molecules, which has two components. One is head and the other is tail. So head is phosphate portion and the tail is the lipid portion, which is usually insoluble and non-polar and hydrophobic. And head is polar and with hydrophilic. So hydrophilic is what? So hydrophobic is which is scared of water or water hating. So cell membrane of prokaryotes is very simple when compared to eukaryotes, which usually contains glycosphingolipids, sphingomyelin, and cholesterol. There are two different types of proteins that are embedded in the membrane: integral proteins and peripheral proteins. So these are integral proteins and the outside one are peripheral proteins. So you can see here integral proteins. These are peripheral proteins. So what is the function of proteins in cell membrane? They are cell adhesion, that is they allow cells to get adhesive to each other, that is they get attached, the two cells can be attached to each other. Then pumps, they act as protein pumps, carriers, ion channels, and receptors. Then the underlying most cell is thin, which has thin layer of fibrils that is collectively made up of basement membrane, or more properly the basal lamina. So below the cell membrane you have a very thin layer which is called as basal lamina. Moving on to mitochondria. Over billions of years, aerobic bacteria were engulfed by eukaryotic cells and evolved into mitochondria. So the functions of mitochondria are regulation of apoptosis and oxidative phosphorylation. Oxidative phosphorylation is a process where glucose is converted into pyruvate. Then mitochondria are sausage-shaped organelles. So they look like sausages. Then the mitochondria has an outer membrane, intermembrane space, and inner membrane, which is folded to form shelves like cristae, and a central matrix space. You can see in the image, you can see the outer membrane, then you have intermembrane space, then an inner membrane which is folded into cristae. So my human mitochondrial DNA is a double-stranded circular molecule. So mitochondria has its own DNA which is of double-stranded and circular molecule, and it consists of 16,500 base pairs. So mitochondria have an ineffective DNA repair system, and the mutation rate for mitochondrial DNA is over 10 times the rate for nuclear DNA. So mitochondria can divide on its own, and it does not require any mechanism that sends messages from the nucleus for cell division or the cell organelles to get divided. So that is the main feature of mitochondria. Moving on to lysosomes. Lysosomes are acidic in nature, and present in the cytoplasm. They have external material called such as endocytosed bacteria, as well as or-not cell components can be digested in lysosomes. So the interior of lysosome is always kept acidic by the action of proton pump or H plus ATPase. So lysosome contains around 40 types of hydrolytic enzymes. These enzymes are all acidic hydrolases. So lysosomes main work is to neutralize or digest the worn out cell components. By keeping the cell fresh and hygiene, lysosomes work in order to digest the worn out components. Moving on to peroxisomes. Peroxisomes are zero point 0.5 micrometer in diameter, are surrounded by a membrane and contain enzymes that can either produce H2O2 oxidases or break it down to catalases. So peroxisomes can break down the component, or they can form compounds like oxidases. So the matrix contains around 40 enzymes which operate in concert with enzymes outside the peroxisomes to catalyze a variety of anabolic and catabolic reactions. So peroxisomes play a vital role in the formation of food, or breaking of the component into various components. So, peroxisomes can form budding of the endoplasmic reticulum or by division. So, peroxisome is obtained from endoplasmic reticulum, or it can divide by its own. Moving on to cytoskeleton. Cytoskeleton maintains the cell structure. It is like fibers, a system of fibers, and it can change the shape of the cell and also help in the movement of the cell. The cytoskeleton is made up of microtubules, intermediate filaments, and microfilaments. So microtubules are long hollow structures with 5 nanometer walls and surrounding a cavity of 15 nanometer in diameter. So there are two globular protein subunits, alpha and beta tubulin. You can see in the diagram there is a blue color and a purple color uh, globular proteins present. There is also a third subunit, which is gamma tubulin, which is associated in the formation of microtubules by the centrosomes. Then they form the they help in the formation of spindle fibers in the mitosis during mitosis. Moving on to intermediate filament, that is also a subdivision under cytoskeleton. Intermediate filaments are 8 to 14 nanometer in diameter and are made up of various subunits. They connect the nuclear membrane to cell membrane, form a flexible scaffold for the cell, and help it resist external pressure, frequently used as cellular markers. Cellular markers are used in gel electrophoresis and various genetic fields. So intermediate filaments are between they are the main chain between the microtubules and microfilaments. Moving on to microfilaments, these are the long solid fibers with 5 to 9 nanometer diameter that are made up of actin. uh, the filamentous actin refers to intact microfilament, and globular actin refers to unpolymerized protein actin subunits. So you can see the picture of a microtubule, then intermediate filament, and then the microfilaments of ectoplasm. Moving on to next slide. Centrosome. So centrosome is close to nucleus and is always present in eukaryotic animal cells only, and made up of two centrioles and surrounding a amorphous pericentriolar material. The centrioles are short cylinders arranged so that they are at right angles to each other. So you can see in the image that the centrioles are short cylinders which are arranged at right angles to each other. Then the microtubules are in a groups of three run longitudinally in walls of each centriole. 9 of these triplets are spaced at regular intervals around the circumference. So you can see there are 9 triplets of the centrioles that are present in the around the circumference of the circle. Then the centrosomes are microtubule organizing centers that contain gamma tubulin. So they contain gamma tubulin protein. Cilia. Cilia are specialized cellular projections that are used by unicellular organisms to propel themselves through liquid, and by multicellular organisms to propel mucus and other substances over the surface of various epithelia. So cilia in unicellular organisms helps in movement, that is, it helps the bacteria to move in the liquid. And in multicellular organisms like humans, it helps in movement of the mucus. You can find cilia in the in the trachea of a human, which help in propelling the mucus and other substance. So within the cilia, that is cilium, there is an axoneme, which comprises of a unique arrangement of 9 outer microtubule, that is, they are doublets, and 2 inner microtubules. So 9 plus 2 arrangement. So this is the arrangement of the cilium. You can see the outermost microtubules, which are doublets, and the 2 inner microtubules, which are 2. So 9 plus 2 arrangement. At the base of these axoneme, there is just inside lies the basal body. Nucleus and related structures. A nucleus is present in all eukaryotic cells that divide. Then the nucleus is largely made up of chromosomes. The structure in the nucleus that carry a complete blueprint of all heritable species and individual characteristics of the animal. So the nucleus consists of genes which carry the character and heritable uh, features of a human. So these nucleus consist of chromosomes, which are made up of giant molecules of DNA. So a DNA strand is 2 meter long. It can fit into a nucleus because at intervals it is wrapped around a core of histone protein to form a nucleosome. So the structure of chromosome is of strings of bead. So you can see these histones and the DNA being wrapped around the histone proteins. So the whole complex and DNA protein is called, the whole thing is called chromatin. The ultimate unit of heredity are the genes on the chromosomes. So the genes on the chromosomes are the heredity units. The nucleus of most cells contain a nucleolus, a patchwork of granules rich in RNA. They are a site of synthesis of ribosomes. The structure in the in the cytoplasm in which proteins are synthesized. So the ribosomes were synthesize the proteins. So the interior of nucleus has a skeleton of fine filaments that are attached to nuclear membrane or envelope which surrounds the nucleus. It contains a nuclear core complex. Each complex has 8-fold symmetry and is made up of about 100 protein organized to form a tunnel through which a transport of protein and mRNA occurs. So there are nuclear pores through which the mRNA is passed from nucleus to the cytoplasm. So the important things in nucleus and nucleus relate the structures in it are the histone proteins, the chromosome, the chromatin, then the genes, what are genes, and the nuclear pore complexes, which are the important parts of nucleus and related structures. Moving on to the next topic, endoplasmic reticulum. Endoplasmic reticulum is a complex series of tubules in the cytoplasm of the cell. The inner limb of its membrane is continuous with the segment of the nuclear membrane. So, in effect, this part of nuclear membrane is a cisternae of endoplasmic reticulum. So, the endoplasmic reticulum starts from the segment of nuclear membrane and moves until the end of the close to the cell membrane. So there are two types of endoplasmic reticulum. One is rough endoplasmic reticulum, which consists of ribosomes on the cytoplasmic side of the membrane, and smooth endoplasmic reticulum are where ribosomes are absent. So so here is the picture of a rough endoplasmic reticulum and smooth endoplasmic reticulum. So the granular endoplasmic reticulum or the rough endoplasmic reticulum is involved in the protein synthesis and the initial folding of the polypeptide chains with the formation of disulfide bond. And the granular endoplasmic reticulum is involved in the steroid synthesis and and is found in the steroid secreting cells and the site of detoxification process in other cells. So the there are two different types of endoplasmic reticulum. One is rough and the other one is the smooth endoplasmic reticulum. A rough endoplasmic reticulum is involved in the protein synthesis, and the smooth endoplasmic is involved in steroid synthesis and detoxification processes. Moving on to ribosomes. Ribosomes in eukaryote measure approximately 22 by 32 nanometer. Each is made up of a large and a small subunit called on the basis of their rates of sedimentation in the ultracentrifugation. The 60S and the 40S subunit contains many different proteins, and at least 3 ribosomal RNAs are seen. It is the site of protein synthesis, and free ribosomes synthesize cytoplasmic proteins such as hemoglobin. So you can see in the picture there is two subunits. One is large subunit, the other one is small subunit, which is 60S and 40S subunits. So protein synthesis is the main function of ribosome. Moving to the next, Golgi apparatus. The Golgi apparatus is a collection of membrane enclosed sac called cisternae that are stacked like dinner plates. You can see there are stacks of the Golgi apparatus, which is a cisternae, arranged one above the other. So one or more Golgi apparatus are present in eukaryotic cell, usually near to the nucleus, and the Golgi is directed at proper glycosylation of proteins and lipids. There are more than 200 enzymes that function to add, remove, or modify the sugars from proteins and lipids in the Golgi apparatus. So when a protein and lipid enters into the cell, the Golgi apparatus main function is to add or remove or modify the sugars that is present in proteins and lipids. So Golgi apparatus is a polarized structure with cis and trans sites. So the membranous vesicle containing newly synthesized proteins bud off from the granular endoplasmic reticulum, fuse with cisternae on the cis side of the apparatus. The trans Golgi vesicle shuttle to the lysosomes and to the cell exterior via constitutive and non-constitutive pathways, both involving exocytosis. Vesicles are pinched off from the cell membrane by endocytosis and pass to the endosomes. So you can see the image, whatever I read now I have put into an image. So the proteins or any complex compounds which is present in rough endoplasmic, that is being synthesized by rough endoplasmic reticulum, is moved in the form of transition vesicles. These transition vesicles go and fuse with the cis phase of the Golgi apparatus. When they fuse with this phase of the Golgi apparatus, it is being either added, removing, or modifying of the sugar is taking place in the Golgi apparatus. After which it buds off from transphase as secretory vesicle and is moved out of the cell membrane in the form of exocytosis. I hope you understood the image of the image that I've put. Please do refer it again or replay the video again to see what is the process from rough endoplasmic reticulum until the exocytosis through the cell membrane. So this ends up the introduction to cell physiology. In the next video we'll be dealing about biochemistry and mainly on carbohydrate. Please do subscribe to the channel to know more about the different chapters that is present in Allied Health Science syllabus, which is purely following Rajiv Gandhi University recommended syllabus. Thank you.